This is the first in a series of video lectures for this week. This week we are going to cover DNA replication, the cell cycle, mitosis, and meiosis. So that's a lot. So there's going to be a whole series of video clips, and it looks like I'm limited to about 10 minutes per video, so that's where I'm going to kind of cut off. So if you notice things get abruptly stopped, that's why. That's my size kind of limit. So here we go. These are what we're expected to know by the end of this section. This is specifically about DNA replication and the cell cycle. There are embedded animations within this PowerPoint, so I am going to let them play, and which is going to add to my time, but that's okay because they're going to start anyway, so I'm going to let them play. In the lab lecture, we talked a little bit about cell division, and we know that when a cell divides, it has to duplicate or copy its DNA first. Once the cell divides, there's 2 new cells, and there needs to be enough DNA for each of those cells to have a complete copy. Now we're going to start using the term genes, and that may or may not be familiar to you, but on the DNA, sections of the DNA code for a certain trait or a certain gene. And so it's going to be important that that full set of information is in each of the new cells. Since DNA controls all of the cellular function or everything that the cell is doing, the DNA replication process has to be very exact. Any mistakes are going to have consequences. So the cell has to be very particular and very precise about that copying process. The reason that this process works and is so specific is because of the law of complementary base pairing. When we look closely at a DNA strand, we're going to look at its structure. And maybe you talked about this when you talked about nucleic acids with Dr. Lin, I'm not exactly sure. But a DNA strand has 2 strands, and it has bases that are complements of each other, or they're little pairs. And so that law, or that base pairing, is what allows new DNA to be an exact copy of the original DNA. And we're going to look at that process closely. DNA replication has 4 steps. I'll be honest, a lot of times I jumble together the first 2 as one step, but okay. You can break it apart into 4 if you really want, if we're really picky, which of course we are. So the first step is the DNA unwinds from histones. Histones are proteins that the DNA is wrapped around. So when we look at a chromosome, it's technically DNA and histones. And we really just say that it's DNA because that's the functional part of a chromosome, but technically it's wrapped around proteins called histones. So DNA unwinds from the histones as the first step. Again, I usually just glaze over that step and jump to step 2. Step 2 is that DNA unzips. So we've already looked at DNA as a double-stranded nucleic acid. It has 2 strands and it makes a helix shape. That means kind of like a spiral. So step 2 means that that spiral is going to unzip or separate. Okay? So the process for this is requires an enzyme called DNA helicase. When you learned about enzymes, I hope you learned that the suffix or the ending of all the enzyme names is ase, or most of them. And so that's how you can remember that DNA helicase is an enzyme. And that enzyme helps to separate or unzip a segment of the DNA molecule. That point of the DNA opening has a special name. It is called the replication fork. Step 3 is the DNA polymerase, another enzyme, builds new DNA strands. We're going to see all of this in the animation. And the polymerase helps add the new nucleotides in order so that the bases are paired up according to the base pair rule. Again, we're going to see this in the animation, but the polymerase moves along the segment of DNA and adds new nucleotides as it goes. This does occur in little sections on one side of the DNA. And so on that side, another enzyme needs to join in, and that is called DNA ligase. This enzyme joins those short segments of DNA into one continuous segment. Ultimately, or at the end, 2 new DNA molecules are made from one original DNA. This is called the semi-conservative replication. That's because each new DNA consists of one old strand or one original strand and one new strand that has just been made with the DNA polymerase. The fourth step, and this is sometimes glazed over as well, but that newly made DNA is repackaged. So the new DNA, this occurs, all these replication forks occur at thousands of different locations on the DNA, and when it's all said and done, that DNA gets kind of repackaged back into 46 chromosomes. All of that occurs in 6 to 8 hours, which is crazy if you think about it. So each new DNA molecule then wraps back around the histones. Okay, here's a visual of what that DNA replication looks like, and we're going to watch the animation as well. So the orange pumpkin-looking enzyme, that's the helicase. That's what opens up and separates the strands of DNA. And then the green blob is that DNA polymerase. It moves along the strand of DNA and moves along in opposite directions, adding new nucleotides. Those new nucleotides are kind of what's floating around in the white space. They look separate, but as they join in on the polymerase, they add in order, keeping the original sequence of the DNA.
Begins at a specific point in the DNA molecule called the
Let me turn this up just a little bit.
origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule